Today we have Dr. Marion Pepper, who is Associate Professor in the Department of Immunology here at the University of Washington, who will talk to us about immune memory to SARS-CoV-2. And before she gets started, we'll have a 5-minute update from Trevor Bedford from the Fred Hutch, who will go over SARS-CoV-2 genomics updates. In terms of questions, go ahead and put your questions in the chat, and I will curate the questions and we'll hold them for the end of the seminar for both Trevor and for Marion. So I'll pass it off to Trevor.
Thank you, Helen. Yeah, and I don't want to take too much time here, but would like to give an update on what we've seen with the latest circulation of variant viruses. So a month ago, talked about the kind of newly emerged 452 viruses, BA4, BA5, BA2.12.1. In the last month, we've really been seeing them spread. So, so if we look at overall diversity, kind of the biggest groups that have been circulating are in the last 6 months are Delta, which is basically dead, Omicron BA1, which is almost dead, and Omicron BA2, which has really been what's been circulating lately. So we can see this pattern here, where if we look across different countries, we can see in purple, let's start with brown with Delta, that this is partitioning cases, confirmed cases by variant proportions. And so we see that Delta's kind of disappeared by February, March, basically everywhere, maybe some places that it's hung on a little bit longer, but it's basically gone from the world now. And if we look at Omicron BA1, which had caused really the large January epidemics in most places, we can see that's been on a steady decline in absolute counts, basically almost everywhere, and in the US included. We're down to maybe just a few hundred a day of cases of Omicron BA1. And then we see on the other hand that BA2 has been causing epidemics in some places slowly, and other places maybe more on the downswing, but basically everything is BA2 now. And so then on top of that BA2 background, we have newly emerged variants. So BA4 and BA5 with 452R in spike and 486V in spike. These appear to be quite important mutations here. There's some uncertainty of exactly this phylogenetic relationship of how this structures, but it doesn't hugely matter. The makeup is the same. And we see BA4, BA5 predominantly in South Africa. It may not be where it actually emerged, but it's where it was first detected. And now most cases in South Africa are BA4, BA5. And then we also have BA2.12.1 emerging from the northeastern US, New York as the main focus there. This has a similar 452 mutation to 452Q, though it lacks other mutations. There's another mutation spike, which doesn't appear as quite as interesting. There's not a lot of mutations going on. We think that all of the fitness would really be driven by this 452Q. And so if we look in the US then, we've seen in the last couple months that really a lot of the increased caseload is due to BA2.12.1, which has been kind of coming along up compared to if you look at the slope here at BA2. Still, the majority of viruses are BA2 rather than BA2.12.1, but it's getting close. And then more recently, we can see BA4, BA5 arriving and really starting to take off in the US. And so if we look at this here, this is the same plot 3 times, but just drawing these lines to get slopes of increase in frequency or fitness. And then we can see that in this case that BA4 has a growth rate of 0.13 per day, BA5 of 0.13 per day, and BA2.12.1 of 0.05 per day. So it really looks like, although this is early data, that BA4 and BA5 appear to be more competitive than 2.12.1. And we can see this as well if we drill into some of the states that actually have data. So the same idea of BA4, BA5, BA2.12.1, and then Georgia, Illinois, New York. And so you can see that across these states that generally the BA4, BA5 are doing better than BA2.12.1. So the expectation here is that over the coming months that we'll see, coming couple months, that we'll see BA4, BA5 displace BA2.12.1. And of course, we could have something else emerge anywhere in the world and really start to take off and spread. It doesn't mean that BA4, BA5 will definitely be the winners. Something else could emerge. And I will stop there and hand it over.
Okay, great. Thanks, Trevor. For those of you who've just joined, we're going to go ahead and hold the questions until the end for both Trevor and Marion. And I'll go ahead and introduce Marion now. So Dr. Pepper is Associate Professor of Immunology at the University of Washington. She is the interim chair of immunology. She completed her PhD at the University of Pennsylvania, looking at CD4 T cells in Toxoplasma gondii, and then postdoced at the University of Minnesota before coming here to the University of Washington. She was awarded the Burroughs Wellcome Investigator Award in 2017, and in the last several months now has collaborated with us and with many others on studies of hybrid immunity to SARS-CoV-2 due to vaccines and repeated infections in people who have experienced infection in the past. So thanks, Marion, for coming to speak to us today.
Yeah, I'm really happy to be here. Let's get this going and hopefully you can see my screen. Yeah. And then the one update I would say is I am now the full-time chair of immunology. So we'll add that. It's really nice to be here. I have really enjoyed my collaborations and discussions about SARS-CoV-2 with Helen and others here at the University of Washington. And I'm just going to share some of our thoughts about how—
Marion, can I interrupt? Yeah. There's like a colors palette that's kind of over the top of your slides. Can you see that on your screen?
No, but that's exciting. Let's see if I can.
Sorry.
No, that's fine. What is it?
Do you see it in the bottom right now? Now do you see color, like this color planet? Yeah, so get rid. Yes, now try again. That was showing up over the slides.
Of color to brighten everyone's morning. Okay, let's see.
It's maraschino. That's nice.
Yes, there we go. Thank you.
Okay, thanks, Trevor. Yeah, so my lab has always focused on immune memory, and we really are interested in how immune memory forms to viruses, to allergens, to all sorts of different things, and comparing the way that different immunological insults, things that the immune system perceives, leads to different types of memory. And what I'm going to talk about is sort of how we've thought about immune memory to SARS-CoV-2 and where we feel like we are in this whole process at this point. So immune memory, as many of you know, emerges when the immune system first encounters a virus or a pathogen of some sort. And the way that the immune system is set up, which is pretty fascinating, is that you need to have cells of the adaptive immune system that can recognize any piece of a pathogen across bacteria, fungi, viruses, all parasites, all sorts of different pathogens. And so the way that the immune system has evolved to have this breadth of responsiveness is to have a very small number of cells that can specifically respond to any piece of a pathogen. So what that means, however, is that when you actually see that piece of a pathogen, that small number of immune cells needs to rapidly proliferate. And so here the virus is in red and just showing sort of viral replication and viral load over time. And the immune system is in blue. And so you start with this very small number of cells that can possibly respond, but then those cells proliferate dramatically, logarithmically. So in mice, you start with maybe 200 cells that can respond to a specific peptide MHC complex, which is what a T cell can bind to and respond to. That number goes up to about 500,000 within 5 or 6 days. The other thing that happens during this time is because you don't want any given immune response to be pre-directed to a specific type of pathogen, these immune cells are kind of blank slates. They don't really have an identity yet. And so during this responsiveness to the virus, these cells not only proliferate, but they also gain these attributes that are specific to fighting off that pathogen that you just saw. So this is a really important time for an immune response, this first 5 to 7 days, this first week of an immune response. And what's remarkable about this is that this is also when immune memory is largely set up. So you not only create cells that can control the virus and hopefully clear that virus down to undetectable levels, but you also create a pool of cells that will persist for hopefully the lifetime of the individual. There is this dramatic contraction period that happens between the peak of this response and when this immune memory sort of gets set up. And about 90% of those cells that you just put a lot of time and energy into dividing and differentiating will die by apoptosis. But what you're left with is this really unique population of cells that have attributes that cells with the same specificities didn't have at the beginning of this response. And what that endows them with is the ability to, instead of taking 5 to 7 days to develop these characteristics, be able to control the infection, these cells, if they see this pathogen again, can now respond within hours. And so if we look at this in a model system where we immunize a mouse and then come back 30 days later and re-challenge that mouse, you can see an immune response within 2 to 3 hours after that pathogen has been perceived. So it's that difference between 5 to 7 days of developing this immune response to just a few hours to a few short days of developing an immune response that prevents disease. And this is the underlying principle of why vaccines work. So my lab has always been really interested in what is unique about these cells. How do these cells acquire these functions? How are they maintained? Where do they go? And how do they control disease so rapidly? And it's really important to understand that there are multiple layers of immune memory, and these layers act in concert to block infection or control viral replication and disease. I think one of the things that we've commented on previously is that so much of immunity is focused on neutralizing antibodies right now, which of course are very important. But what I'm going to talk about is all of the other aspects of immunity that underlie those long-lived plasma cell antibodies. So those neutralizing antibodies that everyone has heard about, I think at this point, which are antibodies that are constitutively expressed in the body that can create basically a force field that can bind to a virus and prevent it from infecting, is the ultimate differentiation state of the immune system. If you can have lots of antibodies around that can constitutively prevent infection, then none of us would ever get a cold. We would never encounter pathogens or infection. It would be ideal. But if you think about how many pathogens you would have to actually create these long-lived plasma cell neutralizing antibodies for, and how pathogens, as we've seen over just this pandemic, evolve, it would be really hard to maintain these really high levels of antibodies that could prevent infection. And the reason why I don't worry about this as much is that underneath this protective force field, this is a cartoon from The Incredibles. You have Violet here making her force field of, let's just draw the analogy and say antibodies. Underneath that incredible force field of antibodies is this whole array of characters that are ready to fight off and prevent disease. And the immune system has really truly evolved to prevent disease and death as opposed to necessarily preventing infection. If you can get a mild infection and be okay, then the immune system has done its job. And so what I'm going to talk about today are some of the cast of characters that lie under that force field of antibody protection from neutralizing antibodies, and how we think that they're functioning in response to SARS-CoV-2. So there's a lot of detail on here. I'm not going to go into this in too much specificity, but what I want you to recognize is that not only are there CD4 T cells and CD8 T cells, which are T cells that can fend off the virus, but there are also memory B cells that emerge during that first 5 to 7 days of the immune response. Some of these, and we're just going to focus on CD4 cells here. Some of these CD4 T cells are really good at activating cells of what we call the innate immune system. These are cells that can phagocytose things, can kill things directly. There are other CD4 T cells that are activated to help B cells. And then out of the B cells, there's this entirely unique and interesting process where these B cells actually can gain mutations in their receptors so that they diversify their receptors and are capable of actually binding different variants of proteins to what they were originally activated upon. So even this activated B cell, once it binds its antigen that it's specific for, it will go interact with a T cell called a T follicular helper cell. Those cells will join together, go into a germinal center, and those B cells will now gain mutations that will be tested out in this germinal center response, leading to a higher level of affinity. So you now can have B cells that are able to bind their antigen better, but you also have a very important diversification of that response. And that allows you to then recognize future variants of those proteins that those B cells were originally activated against. And so what we're going to talk about today is these different populations of cells, CD4 T cells, and then the B cells that emerge out of this germinal center response. And there are two different types of memory B cells that emerge out of that germinal center response. One of these populations of B cells is this really diverse population that I was just describing. And these cells retain a receptor that can bind their pathogen of interest, a piece of their pathogen of interest. And what they do is when they see that pathogen of interest, they rapidly form antibody secreting cells. And that takes about 2 to 3 days for them to get reactivated and start producing large quantities of these antibodies. The other population is the long-lived plasma cells that I mentioned previously. And what's interesting about these cells is they don't really respond as well through their antigen receptor because they downregulate it significantly. But what they do is they continuously produce those antibodies that become that force field that can prevent infection and block infection. So these are some of the cast of characters that I'm going to talk about in the studies that we have done looking at the immune memory response to SARS-CoV-2. And one of the things that I just want to highlight before I get into the actual data is what we know going into this, what we knew going into this pandemic about immune memory, and why we had good faith that the immune system would develop memory that would provide some protection from disease and death. And that's because immune memory cells have characteristics that those naive cells that I was originally telling you about, the cells that first see a virus, don't have. First of all, when you generate these immune memory cells that have gone through this process of differentiation and they've survived this contraction here, these cells can live for very, very long periods of time, many years for some of them. And basically what they do is they stop dividing a lot. They divide very slowly. They stop producing a lot of their effector molecules, but they just circulate around the body, basically perusing for that pathogen again, which then allows them to get reactivated really quickly. So it's this long-lived population of these memory B cells and T cells that are slightly enhanced over what you started with, but functionally very different from what you started with. And this has been shown in many, many papers, but this is a really lovely paper from Rafi Ahmed's lab, where they were looking at the immune response to a live attenuated yellow fever vaccine. And what they did that was really amazing in this paper is they developed tools that we call tetramers, and we'll talk about those in a little bit, that label cells that can specifically respond to a piece of a pathogen. And so these are looking at CD8 T cells, which are one of the two varieties of T cells, and tracking how the emergence of these yellow fever specific cells across these individuals across different time points. So they got a cohort of individuals. They made sure that they all had the right MHC haplotype so they could actually track their CD8 T cell responses. And then remarkably, they gave these individuals not only the vaccine, but a non-toxic way of, they gave them what's called deuterated water, which is a non-toxic way of labeling the cells that are dividing during this period of proliferation. And what that does in effect is identify who emerged during those first 10 to 15 days. And then you can then sample the blood and see how long those cells persist. And what you can't see in here is that really steep expansion and then contraction, because this has already happened at this time point that they're first testing 15 days later. As I showed in that schematic, the expansion and contraction usually happens somewhere between day 10 and 15. But what you can see is if you sample these individuals, and each one of these lines is a different person who participated in these studies, you get these very long-lived pools of these yellow fever vaccine specific CD8 cells that originally had proliferated during this burst of expansion when the vaccine was first seen. And so this is what we would expect with an acute infection, is that you would get the expansion of these cells and then these long-lived stable populations. And these are CD8 T cells with CD4 T cells. Our group and others have shown that there are similar kinetics, albeit they decline a little bit more quickly than the CD8 T cells. The other thing that's really unique about immune memory cells is even if you can't see them making something constitutively, it doesn't mean that they're not poised to do so. And during this period of differentiation, these B cells and T cells gain epigenetic modifications that basically poise them to respond very rapidly. Some of these cells retain transcripts of the cytokines that they're going to express, so they can just very rapidly translate those transcripts to create cytokines. But this allows for these cells to basically go as soon as they see their pathogen of interest again. And then lastly, these cells don't just circulate throughout the body, they also migrate and they take what we call residency. They move into different tissues throughout the body and different regions of the body. So if you have a respiratory infection, one of the things that will happen is your memory cells, some of those memory cells will actually go back into the lungs where the virus was first perceived and live there as a sentinel population for the life of the host. And of course, what that does is then position the cells right at the entrance of the pathogen. So now you have an expanded pool that has the right epigenetics to rapidly produce cytokines and effector molecules right at the entrance of the pathogen. And it's all of these things together that immunologists have been studying for a long time that we now know endow these immune memory cells with the capacity to prevent disease. And so when the pandemic first emerged, we decided that we could use tools that we've generated in the lab to start to dissect how these different populations of cells were either being formed or not. And we wanted to, of course, look at long-lived plasma cell-derived antibody, as it's important for protection against infection. But we really wanted to look at the cellular response. We wanted to look at the memory B cells, and we specifically knew that memory B cells that had class switched to this isotype called IgG were good at fighting off viruses. We also wanted to look at these memory CD4 cells, both the memory CD4 cells that we knew would be important for reactivating memory B cells, as well as CD4 cells that we knew could make these proteins that are that can control pathogens called cytokines. and activate antiviral innate immune responses. And lastly, we also wanted to look at memory CD8 T cells. And so we developed a cohort of individuals who were very early in the response. They'd been infected in late January and February, mid-February for most of these individuals, that had recovered from mild COVID-19. And based on those kinetics from the Ahmed study that I showed you before, we determined that if we actually knew when people were infected by their PCR positivity test, if we went approximately a month out from that positive test, we should be able to find memory cells that are SARS-CoV-2 specific that were quiescent and looked like they were going to last. And the only way to do that is to have a second blood draw where you can then determine the kinetics of decline between that first visit and that second visit. And we compared these immune responses to individuals who had blood taken first before pre-2019 so that we could actually make sure that we weren't detecting any antibodies in the serum. And we used that as a cutoff for our healthy controls, who at the time, there wasn't as much exposure to infection and there were no vaccines. So we had 17 healthy controls that we used for this study as well that came in at these 2 time points. And what we could immediately show is when we looked at 35 days, approximately 1 month after infection, the vast majority of inflammation had already been resolved. So the blood of people who had SARS-CoV-2 did not look any different from the blood of the healthy controls at this point. And keep in mind, for everyone that we were looking at individuals who'd recovered from mild COVID-19. So none of our cohort was hospitalized. They were symptomatic, but they didn't have severe disease. So it was good to first see that just doing an overall panel of immune correlates, these individuals looked like they had controlled infection and were now in a period of resolution. And so what we did at the time, which was unique at the time, but now is very common, is we generated tetramers that we call B-cell tetramers that have a piece of the SARS-CoV-2 spike that has been biotinylated that we can then attach to a brightly fluorescently labeled molecule here called streptavidin. The fluorophore is called phycoerythrin. And what this does is it lets any B cell that's specific, that has a specific receptor for that piece of the spike protein, to be identified with this fluorescent label. The fact that there are 4 of these adds some avidity to this process, so we can pull out a variety of affinities of B cells. And we made these tetramers so that they either contained just the RBD portion, which is the receptor binding domain of the spike, or we made some that were the whole spike. Because what we knew is that the RBD portion of the spike was going to be critically important for binding to the ACE2 receptor that would allow viral entry. We do this slightly differently than other people. We know that B cells are also really good at binding all the rest of these components of our reagents. They can bind this phycoerythrin molecule at very high levels. They're about 20,000 B cells just circulating around in a mouse that can bind this PE molecule. So what we do is come in with this other decoy reagent first to saturate any B cells that are binding our reagent, not our protein of interest, and then come in with our tetramers that have just a different protein on them, that just have the RBD. This has an irrelevant protein on it, and only 1 fluorophore, whereas this one has an additional fluorophore that lets us distinguish them. And if we hadn't done this for years and years, I would say this looks like a smattering of dots and it's not super convincing, but we've done this for a long time and we know that this is true through a lot of experimental controls, and I'll show you some. But basically what we could do is take the blood of individuals and stain it for both the decoy reagent and our RBD tetramer. either in healthy controls across the top or individuals who'd had the infection across the bottom. This is looking at the RBD specific tetramer. And what you can see in healthy controls in blue is that there's no real increase in these cells. What we know about the cells that we can find here, it's not that they're not real. Those are real. Those are just predominantly naive B cells that are circulating through the blood that have never been activated. And we show that in the paper. But in individuals who'd had infection, already by that 30-day blood draw, you can see enhanced numbers of these RBD specific cells. And if we wait till a couple of months later, you can see that that number goes up. And that's the process of those germinal centers pumping out those memory B cells over time. And so we looked at all of the other parameters that I mentioned before and described. We looked at CD4 T follicular helper cells, which are the B cell helping subset, and CD4 effector T cells. And we found that all of those cells were there. And this was published, this came out on bioRxiv in August of 2020. It was an enormous collaboration across the UW and Benaroya Research Institute. And then what we could see is if we compared healthy controls across all of these different parameters that I just described versus individuals who'd had COVID-2, red means a response above baseline, blue means at the baseline. And you can see there's some differences in how people responded. This person's super interesting. They had a very low B cell response, but a very high CD8 T cell response. In other individuals, you could see higher B cell responses and lower T cell responses. You can see that here in patient 10. And so there was some heterogeneity, but what we were really encouraged to see at the time is that every person that we looked at who had mild COVID-19 was mounting a strong immune memory response, and that was great. And so, of course, the pandemic kept going and vaccines came out, and there were all sorts of other ways that we could start to investigate what immune memory does in people who had previously been infected versus those who were not. And so we started this very exciting collaboration with Helen and her group, in which we decided to compare how our previously infected individuals were responding to vaccination in comparison to people who had not been infected with the original strains of SARS-CoV-2 and had just been vaccinated. And as many of you know, the vaccine schedule, there were 2 vaccine doses. These were all mRNA vaccines for this study, given 4 weeks apart, which is labeled here as V1 and V2. And we had blood samples from our individuals right before the first series of vaccines, 1 week after that second vaccine, 3 months after, and 6 months after, to watch how the immune response waned. And then what was really exciting for us is we were also able to look at how these cells responded to a boost. And so 6 months later, when individuals were starting to get boosted, we were also able to look at how that influenced the immune response and really ask the question of whether a prior infection altered your vaccine response. And we assumed that it would, but it wasn't entirely clear what the parameters would be. Would it alter the quantity of the number of cells that you had that were specific for the virus? Would it alter the quality? Would these vaccinations basically normalize everything that had previously been generated during the infection? So these were the type of questions that we wanted to look into. And so we did similar types of studies. We basically looked at the quantity of the humoral immune response, looking in people who hadn't been infected versus people who had been infected. And it's almost easier. This is our same strategy of looking at a decoy versus an RBD specific tetramer. And it's almost easier just to watch how the kinetics unfold in previously uninfected people in blue, and then in previously infected people in red. And I think what's interesting, and just in keeping with everything we expected, is People who had been previously infected but not vaccinated, of course, had more of these RBD specific B cells than those who hadn't seen the infection and hadn't been yet vaccinated. But it's not an enormous difference in cell number. And again, when you think about that schematic that I drew, where 90% of the memory, 90% of the antigen specific cells contract, and you're left with 10% greater cell numbers of antigen specific memory cells, this is in keeping with that. But what was really encouraging is even after just those 2 vaccines, people who were previously infected were boosted to really high levels. People who were not previously infected were mounting a significant immune response that didn't quite catch up. But you could normalize the actual numbers of RBD specific B cells by adding in additional doses for the individuals who hadn't been infected. Now, what we saw that was interesting, and we still sort of need to tease apart, is the individuals who'd been previously infected always retained slightly higher levels of these memory B cells at these later time points. And we haven't done the full analysis of what a later time point would look like. This is just 2 weeks after the second boost. So we're still waiting to see if there is a numerical advantage for these individuals who'd been previously infected. And that might be because of those ongoing germinal centers that lasted for many months in between infection and vaccination and this boost here. So we're still looking to see what happens there. You can see the exact same pattern in the antibodies that are made by these individuals. Again, you have antibodies that persist in people who've been previously infected. Those get massively boosted after vaccination. And those are retained at higher levels, especially out to 6 months post-vaccination, where we had the serum and could look at this. Whereas individuals who were just vaccinated lose that same level of antibody production. And I think this is really interesting and really raises a question about whether the vaccines are doing as good a job at generating long-lived plasma cells as an infection, which it is hard to say at this point. And we're going to have to look at later time points, but it does seem like there is a more rapid decline of antibodies after vaccination than after vaccination plus, infection plus vaccination, which you can see here. And so if we looked at the T cell quantity, we did this by stimulating cells with different pools of peptides and then looking at a couple of different parameters to see how they responded. We could look at the upregulation of markers of activation, just to say that these cells have bound a peptide through their T cell receptor and become activated. We could also look at the expression of cytokines, which would show what their functional value is. What are they actually doing when they see this antigen again? And just to show the stringency of this assay, what we decided to do was to look both at peptides that were only present in the virus, so from the M and N proteins that are in the virus but are not in the vaccine, as well as peptides that are in the vaccine as well as the infection. And what you can see here is in individuals who were not infected at all in our healthy controls, basically, there is no real presence of these M and N specific cells as you would expect, because again, these M and N peptides are in the virus, they're not in the vaccine. But you can see when those individuals were vaccinated, the pool of cells that's very small before vaccine that's capable of responding to spike goes up significantly. And so these are now spike specific cells that you can see in vaccinated people. In people who were infected, we can see these M and N specific cells, again, supporting the rigor of these studies and demonstrating that they definitely had been infected. And with their spike responses, you can see that these go up with vaccination too. And then this is just shown graphically, showing the stable low levels of M and N specific cells in individuals who weren't infected versus those who were infected, this stable ongoing population that doesn't really change over time, and doesn't get reactivated by the vaccine in any sort of antigen non-specific way, which is good to see. And then here are the spike numbers. And what this shows you is that individuals who were vaccinated rapidly gain enough CD4 T cells in this assay to resemble what we saw in the people who were previously infected and then vaccinated. So the vaccine does a very good job of inducing high numbers of CD4 T cells that are comparable to those seen in previously infected individuals. We can do this with different types of memory B cells. You see a similar thing. The quantity of the RBD specific cells that are truly memory cells can be normalized with vaccination and these 3 antigen doses in these previously uninfected people. So it did seem like, at least for the numbers of cells that we could find, that they could be comparable between vaccinated and previously infected vaccinated individuals. But when we looked at the quality of the response, and this was something we were really interested in, we noticed a difference. And there had been a lot of studies that were coming out that were suggesting this idea of hybrid immunity, in which if you'd had a previous infection prior to vaccination, you were better protected than either being just infected previously or being just vaccinated. And what we could see here is this is now we've taken antibodies from these people at 6 months post-vax 2. Again, blue is naive, red is previously infected, or 2 weeks post-vax 3. And we asked the question, do the serum antibodies that are present at this time point, which are probably those antibodies from the long-lived plasma cells, are they capable of responding to variants? And at the time, and Trevor showed this nicely at the beginning, Delta was a variant of concern. And what we could see is if we took the serum from individuals who had been vaccinated 6 months before and just looked to see if that serum was neutralizing against Delta, it really wasn't. But that boost generated a burst of activity from the memory B cells that were quiescently circulating through these individuals that allowed them to produce those antibodies that were then capable of neutralizing Delta at the same level that people who had been previously vaccinated. So that was exciting for us, that the boost could actually reactivate those memory B cells so that they had comparable antibodies to people who'd been previously infected. And we did the same thing with Omicron, and you saw the exact same pattern. And what was interesting about these data is it actually showed us 2 things. Number one, in previously uninfected people, that boost was really creating a burst of antibodies that was going to protect against variants. Unfortunately, we don't think it lasts for terribly long, but we're still looking into that. But the other thing that was interesting to us is that in people who were previously infected, not only did they already have this breadth of antibody response, suggesting that their long-lived plasma cells were doing a lot more than in the people who were previously vaccinated, but that the boost didn't really make much of a difference in their antibody titers and their neutralizing antibody titers, because they'd probably already hit this peak. So there was a lot to sort of pull out from that. The other thing that we looked at, and I'll just point you towards these lines because I think it's the easiest way to look at this. We looked at cytokine production between previously infected individuals in red and just vaccinated individuals in blue. And even though we had seen the same numbers of responsive cells from individuals who were previously infected and had this hybrid immunity that comes from infection plus vaccination, so you need this vaccine to boost this up, they had higher levels of interferon gamma at every one of these time points that we looked at that couldn't be recapitulated by multiple doses of vaccine. So there's something that is inherently different between the cells that had perceived infection during that first 5 days or 10 days of differentiation versus those that had perceived the vaccine. And we also saw this with another cytokine, interleukin 10, And this one was particularly interesting to us because interleukin 10 does a lot of different things, but one of the things that it can do is actually dampen an immune response and decrease pathology. And so individuals who had been previously infected had this added component of these higher levels of IL-10 producing cells. So even though the numbers were the same, both the quality of the antibodies that were generated as well as the qualities of the cytokine producing T cells were different if you'd been previously infected and vaccinated versus just vaccinated. And that's just depicted here graphically. We could normalize a lot of the different immune responses through multiple doses of vaccine, but not all of them. And especially the CD4 T cell cytokines really retained these imprinted characteristics from whatever those cells saw when they first saw the virus. And so I just want to go back to this idea. There's a lot right now being highlighted in the news of reinfections and how the immune response isn't working the way scientists thought it was going to work. And there was just this article yesterday that was highlighting, how often can you be infected with a coronavirus, and the spread of the Omicron variant has given scientists an unsettling answer. And I would argue vehemently against that. I think what we see in our studies and just in the epidemiological studies, just looking at the case numbers versus hospitalized people versus deaths, is that the immune system is actually working exactly the way we would expect it to. It's fighting a mutating virus, but thankfully we have all of these underlying T cells and B cells that are able to recognize variants and control them before severe disease sets in, which is actually how the immune system has evolved. It's evolved to keep you alive and is working actually really well. I think watching these memory B cells kick in, and especially with the variant data, what you're seeing here in this giant boost of antibody versus either Delta or Omicron is those underlying memory B cells that don't constitutively make plasma neutralizing antibodies, but they will upon reactivation, and they'll do it rapidly enough to help control that disease. And this is exactly what the T cells are doing as well. So I think there's it's buried in the article that the good news is that most people who are reinfected with new versions of Omicron will not become seriously ill. I would like to say that that is what our immune system has evolved to do, is to not actually be fully sidestepped by a variant. And so I think it actually isn't unsettling what we're seeing. I think what we're seeing is exactly in keeping with what we would think would happen with the immune system. And so with that, I just want to thank the people who've done this work. Lauren Rada was a lead on both of these studies with Jason Netland. We were at an immune memory meeting in Banff when we first decided to take on these studies. And so that picture is from there. Those first studies were a huge collaboration with David Rawlings' group at Seattle Children's, the Gale Lab, the King Lab, the Institute for Protein Design, people at Benaroya Research Institute. David Wiesler gave us some proteins. And then the second half of this was continued with a lot of help from Helen and these three in the lead, Kurt Pruner and Peter Morawski from the BRI, who did the T cell studies, and Lauren did the B cell studies, as well as our funding and our volunteers. So with that, I'd happily take any questions.
Great. Thank you, Marion. That was wonderful. That was such a nice overview of the work that you've done over the last 2 years, and represents so much progress in what we understand. I'm going to go ahead and start with the questions in the chat, and then I'll I think Deb has the question, and then feel free to go ahead and raise your hand, and I'll call on you one by one. And if we don't get to your questions, I'm sure Marion would be happy to have you email her. Okay, so there's a question from David Gordon, which is, if it only takes about 5 to 7 days for immune cells to proliferate, why does the CDC say that you have nearly no immunity 7 days after a COVID vaccine? I think that's a question for you, Marion.
I would disagree that you have no immunity at 5 to 7 days. I think it's emerging though. I mean, it takes time. It's a rough estimate, 5 to 7 days. Some people may have it a little earlier. Other people might have it a little later, but I think that to say it's no immunity is inaccurate. It's just emerging immunity. And so to be safe, saying that you're going to go beyond 5 to 7 days is just safer.
Great. The next question is from Kelly Lee, and really about the question of the order of events. Is the response for the infected then vaccinated the same as those who are vaccinated and then infected? Getting at the question.
Yeah, it's such a great question. These studies are ongoing right now in my lab, and we're just getting those started. The other studies are emerging from other labs. The Wiesler lab, Laura Walker's lab, I'm forgetting others that are emerging right now. It's looking interesting. I'm not sure we're seeing exactly the same things, and I don't think we quite know if that difference in the T cell functionality is going to be maintained. So the answer is they're ongoing studies, and we just don't know yet. But I think it's important because a lot of us have been infected after being vaccinated. And I'd love to think that we've gained some of these attributes.
Right. But those, I think a lot of those publications are about to come out. So
Yeah.
we need to see what
They're coming.
Yes. I'm going to turn it over to Deb to ask her question, and then I'll go back to the chat.
Okay, great. That was a great talk. Really enjoyed that. I had a couple questions. Actually, that last question was one of them, but the other one I have is just a simple question. Do you think that there's any merit or in updating the vaccine, given that you're seeing really nice development of a broad repertoire of neutralizing antibodies, especially with the hybrid immunity? What's the merits of really updating the vaccine? That's number one. And then number two, I want to ask you whether or not you think, because you touched a little bit on this, on immune imprinting, kind of the phenomenon that has been discussed a little bit with influenza. Is there a potential that there, once immunity wanes and then you get exposed to a new variant, could there be sort of competition between the memory cells such that whatever you were exposed to first ends up preferentially recalling and, really potentially dampening your protection from that new variant?
Yeah, those are both really good questions. So as far as the question about, that's what I was going.
Updating vaccines.
Yeah, updating vaccines. Yes. So I personally think that we should update vaccines. It's hard to say, do we start now? And is that going to be relevant in 4 months? Probably not. But I think anything that can diversify the response would be good, because yes, there is significant amount of cross-reactivity. People are protected and have antibodies that can neutralize variants if you have this hybrid immunity. But I think having additional exposure to those altered regions of those proteins is not going to be a bad thing, because you're already going to have memory B cells that can hopefully respond, and you might be able to increase their number. But if you don't have those cells there and can pull in new cells into the response, it can only be beneficial. So to me, I don't think there's going to be a lot that we gain by continuously giving homologous boosts. We know that these increases in antibody levels are not only minimal. I mean, we didn't even see a difference really here in neutralization, but on top of that, they're short-lived. And so I think we need to think about introducing other proteins, other variants into our strategies right now. The other thing I think we need to think about introducing is not just diversifying the antigens, but also the route of antigen of vaccine. And I think there are a lot of companies now thinking about intranasal vaccines or even oral vaccines that may not alter the breadth of the response as much, but at least will alter the positioning of these cells and hopefully create more of those sentinels at the mucosal sites. So I think that would be another way to do it. Your second question, is there going to be competition between those memory cells? This is a really tough question. And I think, this idea of original antigenic sin, it's this nebulous concept that incorporates just the fundamental mechanisms of how immune memory works. If you have a pool of memory cells there that can get activated at high affinity, they're going to get activated. And that's not necessarily going to inhibit the activation of cells that could respond to something else, but you just may not see it as much. So I think it's really hard to say at this point that whether there is evidence of that. So there are a lot of us thinking deeply about how to get at these studies and using different animal models to do this, because I think these are really important questions. But I don't think we have the answer to that, unfortunately, at this point.
Great, thanks. Great talk.
Okay, Marion, you have a lot of questions and we probably only have time for one more. So I'm just going to pick the one that I think 5 people have asked, which is about exhaustion, immune exhaustion, and whether or not what your thoughts are on that. And then I will summarize the rest of the questions and send them to you.
Sure. So immune exhaustion is this concept that came mostly from chronic viral infection originally, and then has been predominant in the cancer field. But it's this idea that if the immune system gets activated over and over and over in this sort of chronic exposure to antigens, so the ligands for these immune receptors, that it makes these cells dysfunctional. And I would say that these are not the types of responses that are going to drive exhaustion. These are short-lived periodic bursts of responses, as opposed to these ongoing chronic exposures to antigen. So far, I don't think there's good evidence for any induction of immune exhaustion through infection or through multiple boosts. So I think so far the argument is that that is not part of what we're seeing.
Great. Thank you so much to both of you for coming to speak with all of us today. I think I have learned so much, and I think everyone has been really, just by the quality and the number of questions, I think really got a lot out of both of your talks. I just wanted to mention that next month we'll have Jesse Bloom come and speak, and that will be on Tuesday, June 21st at 9 a.m. And again, Trevor will give a 5-minute update. And thank you to everyone for coming. Please let us know if you have any feedback on the seminar series.